Okay, you guys have been learning about the different types of mutations that people can have. So far, you've just learned about genetic mutations like insertions, deletions, and base substitutions. Um, and each time you learn about genetic disease, we've just been focusing on people. You guys are going to see a few more of these, and I'm actually going to do one where you get to see some genetic mutations for things like horses and dogs. But in this lecture, I want to focus on human chromosomal mutations. So your essential question is, what are the 5 chromosomal mutations? So as you've seen, there are 2 main types of mutations. There's a chromosomal mutation, which we're going to learn about now, and there are gene mutations. Those are things like insertions and deletions and base pair substitutions that you've already learned about. Let's just refresh what a chromosome is. So humans have 23 pairs of chromosomes, with 1 chromosome coming from each parent. The chromosomes are coiled up DNA, and under normal conditions, all of the chromosomes are inherited intact. So this right here is what we call a normal karyotype of a human chromosome. Remember, a karyotype is a picture of chromosomes, and we, as humans, line them all up and number them. So these are pairs 1 through 22, and this last set down here are the sex chromosomes, X and Y. Not all animals have 23, or I should say organisms, 23 pairs of chromosomes. That's just people. So, chromosomal mutations. This is any change in the structure or number of chromosomes. The big difference here between genetic mutations is the fact that these tend to be really large scale, and they affect many genes. When you have a genetic mutation, it just affects one specific gene. In a chromosomal mutation, you're talking about many different genes involved because these are huge, huge mutations that often involves many different genes or entire chromosomes. There are 5 types, and we'll talk about each type. Deletion, duplication, inversion, translocation, and non-disjunction. So a chromosomal deletion is exactly like it sounds. Basically, over here, we have our normal chromosome that's fully intact. So let's say that each letter represents a different gene. So here we have genes A, B, C, D, E, F, and G. Well, if something happens and we have a deletion, so let's say we're going to delete genes C and D, we'll only be left with genes A, B, E, F, and G. So we've deleted 2 genes from a chromosome. Some examples are Wolff-Hirschhorn syndrome. This causes severe mental impairment, as well as Cri-du-Chat syndrome, which causes mewing sounds, and again, extreme mental impairment. I mean, we've lost 2 entire genes. It just, the severity and the impairment can also depend on which 2 genes we've lost and on which chromosome. All right, so in this case, this is a chromosomal duplication. So instead of deletion, we're adding genes. So here, a segment of genes is copied twice and added to the chromosome. So our normal chromosome is on the left. It's A, B, C, D, E, F, G. These are all the different genes. And right here, where the little red arrow is, we're going to have a duplication. So we've duplicated genes C and D. So that means we have extra genes in there. And your body doesn't know that these are extras. So it just turns them on, makes the proteins, and that even excess protein in your body, or specific types of proteins, can cause issues. So this one is Charcot-Marie-Tooth disease, which is a high arched foot, claw feet, and you tend to be confined to a wheelchair. All right, this one seems complicated at first, but it's really not. Basically, it's a chromosomal inversion, okay? So here on the left again, normal gene, A, B, C, D, E, F, G, and we invert it. So basically, we take genes C, D, and E, and we flip them. So instead of C following B, we have E follow B. Now, this can cause huge problems. It has to do with really in-depth molecular genetics and how genes are turned on and turned off, and different transcription factors, and it's not something we're going to touch in this class because it gets really, really confusing in a big old hurry. It's cool to learn about, but we just don't have time to explain it. So basically what happens if we've taken our segment of genes here and turned it upside down. An example of a disease that is related to this is called four-ring syndrome. You end up with a cleft palate, so that's where your palate is the roof of your mouth, and quite often the little flap of skin right underneath your nose is open, and sometimes that palate in the top of your mouth can be open as well, which can cause problems for young babies when they're trying to nurse. It can also cause club feet, so your feet don't resemble a normal foot, and the testes don't descend. All right, next one is a chromosomal translocation. Basically, material is swapped with another chromosome. So let's say we get two chromosomes lined up next to each other, okay? And all of a sudden they swap their genes. And they can swap whole segments, part segments. This can get to be a problem if we have a gene that's split in half, so it doesn't stay in its entirety. Again, this gets to be really cool, and that has to do with that junk DNA. If you watch the epigenetics video, there's some really cool higher level science being done to investigate like what happens with translocations. Sometimes it can be bad, and actually sometimes it can be beneficial because it can actually cause new genes to arise. An example of this happening is Burkitt's lymphoma. It's a cancer of the lymph nodes, and it's quite often seen in children. All right, non-disjunction is the last of the 5. This is a huge, huge mutation. Basically, our chromosomes fail to separate during meiosis. So you remember in meiosis and mitosis, the chromosomes line up down the center line, and they get pulled to each side of the cell splits. This happens once in mitosis and twice in meiosis. So this happens during, it can happen in meiosis 1 or meiosis 2, because remember in meiosis we have 4 divisions, and the non-disjunction can either happen in meiosis 1 or meiosis 2. Okay, so non-disjunction produces gametes, and therefore a baby with one missing chromosome or one extra chromosome. So this right here is an example of non-disjunction. On the right, we only have one chromosome, so it's called monosomy, mono meaning one. And on the left, we have 1, 2, 3 copies of a chromosome. This is called trisomy, tri meaning 3. So non-disjunction and meiosis. Okay, here are the chromosomes, they're all lined up. And in meiosis 1, so this is our first cell division right here, they split normally, okay? So I get one chromosome over here and one chromosome over here, okay? Now, this is meiosis 2. They're going to split again because we're creating gametes, okay? We need a haploid cell. So this chromosome right here does its job correctly. It splits one chromosome here and one chromosome here. The little blue guy on the right during meiosis 2 does not perform the splitting correctly, okay? Basically what happens is, for some reason or another, this little cell over here ends up with no chromosome, and this little cell over here on the left with the 2 blue chromosomes ends up with both copies. This is going to be a problem because when the sperm comes in and fertilizes, the sperm also has 1 copy of this gene. If it fertilizes this chromosome, this egg here with 2 copies of the chromosome, we'll end up with 3 copies of the same chromosome. And if the sperm fertilizes this egg right here that's empty, you're only going to have 1 copy of that chromosome. So again, here's the fertilization. We have the egg with 2 copies of the chromosome. We have the sperm who has its own copy of the same chromosome. They team up to make fertilize an egg, and all of a sudden you have a trisomic zygote. So you have an egg that's fertilized with 3 copies of the same chromosome. So, non-disjunction. Every cell in that baby's body will have 3 copies of this chromosome instead of 2. It's only supposed to have 2. This condition is called trisomy. An example of trisomy that we see is trisomy 21. The individual has 3 copies of the chromosome number 21. That's why we call it trisomy 21. 3 chromosomes of number 21. Chromosomal mutations, most chromosomal mutations are lethal. There are very, very few exceptions to this rule. If the fetus survives, it tends to cause widespread abnormalities. So a really good example of this is Down syndrome. The cause of Down syndrome is non-disjunction of chromosome 21. People who have Down syndrome have 3 copies of chromosome number 21. There's been a lot of research as to why people with Down syndrome have slower development, and what chromosome 21 does in general is it turns out chromosome 21 is actually involved in a lot of your mental development pathways. So you automatically think, well, an extra chromosome would make you develop faster. Well, actually, it turns out not to be the case. Having that extra chromosome in there kind of gets in the way of the mental development, the neurological pathways, and it actually slows things down. This is a karyotype of someone with trisomy 21. So you can see here we have chromosomes 1 through 20, and we only have 2 copies of each. But when we get down here to 21, you can see that there are 3 copies of chromosome 21. And you can see this individual was female because there are 2 X chromosomes. These days, there is genetic screening for a lot of genetic diseases, not only chromosomal mutations, but things like cystic fibrosis. The mother can have an amniocentesis. Basically, what happens is they stick, I'm not going to lie, it's a very large needle into the mother's stomach, and they pull out amniotic fluid. They can then take that amniotic fluid and grow it in culture and basically do genetic sequencing on it to see, hey, does the child have Down syndrome or any other lethal family diseases? So the key point here is too much or too little DNA is bad. All right, and I'm going to back this up a little bit, guys, because I want to talk to you about something. Yeah, ignore that. Okay, so I want to talk to you about this guy right here. So we'd end up with trisomy, and in this one, we'd end up with monosomy. There we go. This is what I want. The majority, there is one known exception where the monosomy is not lethal, and that's something called Turner syndrome, which I'll have in one of the genetic lesson reviews for the diseases. All other versions of monosomy are lethal. The fetus does not survive. So you can see that non-disjunction mutations are pretty bad. All right, scroll back to the down, all the way down. Just ignore that. Go, 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 go. Okay, did that. Okay, so in the slideshow, let's review. What are the 4 types of chromosomal aberrations? There were 5, so you guys should know this. The last one is non-disjunction. And which type of aberration is probably the most damaging? So these are 2 questions you guys need to think about, as well as the essential question. Okay, I will see you guys next time. Adios.